Caroline Uhler at the MIT and Broad Institute, and today she will tell us about multimodal data integration from biomarkers to mechanisms.
So thank you so much for this invitation. It has been a really exciting workshop so far. So I'd like to tell you a little bit about our thinking and some of the results around the tight link between mechanical state of a cell or cell shape, and then chromatin state of a cell, and then gene expression, gene regulation. And a lot of this work is in collaboration with a long-term collaborator, G.B. Shivashankar's lab on the experimental side at ETH Zurich. He showed in very early experiments that if you take the same cell and you just plate it onto different shapes, you get very different gene expression programs out. So that, and then he also hypothesized that, well, what could be the integrator is chromatin organization, because that's changed with different cell shapes or mechanical states. And then also that, of course, has an impact on gene expression, because different genes that come close in space can turn on very different gene regulatory programs downstream. And so we have been, in our group, very excited about thinking about, chromatin state as an integrator where we can really measure these different transcriptional states of a cell, but then also maybe the mechanical state of a cell. And so some of the earliest experiments we did were very simple, where we just look at, the cheapest way of measuring chromatin state of a cell, just DAPI or Hoechst stains. And we saw that, even just using DAPI, it's very, very easy to distinguish between different breast cancer cell lines. And in fact, you can even go much further to differentiate not just different cell lines, different cell types, but actually even same, say, cell line, but just like a little bit of a signal added to it or not added to it. So different cell states. You see here if we add TNF-alpha or not add TNF-alpha. And, we were very interested to know whether this is really what the measurement we need is just chromatin compaction states and not just, things that are what you usually take out of DAPI is just a nucleus shape. So here we literally just take little patches within the cell nucleus so that we're really just measuring chromatin compaction differences. And you see that that's really what differentiates these different cell states. So we're wondering, how far can we push it, right? How much does the chromatin state of a cell that we can measure so easily and so cheaply here, how much does that represent the transcriptional state of a cell? How much does that represent maybe even protein localization within a cell? How much maybe does that even relate to what the genetic background or polygenic risk score is of the person whose cells you're taking? And maybe how much can you just from looking at these cellular images here be able to tell what's wrong in the cell? What was perturbed in the cell? So kind of the reverse question. So those are the types of questions that I want to talk about here in this talk. So how do we even connect things like these images to other single cell measurements, like say single cell RNA-seq, which are such different types of measurements? So early on, 2019, we introduced these multimodal autoencoders into the biological field, really providing a mechanism of how you can integrate different modalities that are never measured in the same cell, like a DAPI stain and single cell RNA-seq. And you actually saw an application of this type of framework in Aviv's talk yesterday, where she integrated OPS and Perturb-seq. So how does it work, right? How can we translate or integrate, say, DAPI staining and single-cell RNA-seq, where there is no clear connection between the two? I cannot say this pixel relates to this gene, right? Or, I can also not measure it in the same cell, so I don't really have training data to do that. But here's the idea how you do it. So say you take a population of cells. My next example is going to be in CD4 T cells. So you take the same population of cells, and I at random take out some for imaging, and I at random take out some for single-cell RNA-seq. Well, I know it's the same population of cells. So if I integrate them into the same latent space, what I'm going to do is I'm going to match the distribution, right? It's the same population of cells. So if I add say, another loss term in the latent space that tells me, well, I have a good representation if I cannot tell whether this cell comes from imaging land or sequencing land. That's good, right? Because the cells were chosen at random for imaging and at random for sequencing. I shouldn't be able to tell whether it comes from sequencing land or imaging land. So that's how you can actually make these integrated latent spaces where you can then use the latent space to take a DAPI-stained image and translate it to single-cell RNA-seq, or the other way around, if we're more interested in this one direction. Okay, by kind of matching distributions in the latent space, even though I've never measured the same cell, I've never measured the two modalities in the same cell. So that's what we're always using, and we have a lot of theory around, how to do this right. These are the models that we've introduced there and that are now, quite widely used. So let me show you the first application that we did of this type of framework and CD4 T cells, as I told you. So we started with, an observation that Shiva's lab had early on, where he had looked at chromatin-stained images in the mouse. And he saw that in CD4 plus naive T cells, you actually have two populations of cells. Some that have heterochromatin peripherally, and some that have heterochromatin centrally. And in addition, he also saw that, these T cells with heterochromatin centrally are softer. And they seem to also have, a functional relevance to have heterochromatin in the center, because the softness also means that you can, transverse quicker, and you also show accelerated activation. So what we did here is we wondered, well, first of all, is this the same in humans? So we just repeated the experiment. So took CD4 T cells in humans. Again, if you just cluster the images, you see very clearly two populations, some that have heterochromatin outside, some that have more heterochromatin in the center. Now, because this framework where we can translate between these chromatin images and single-cell RNA-seq, we wondered whether, what do we see, right? What is the transcriptional profile of these two different populations of cells? Now, interestingly, so here is single-cell RNA-seq data. So if you take, again, the naive population, which is this one here, you just do clustering, you very clearly see two populations, okay? Now, we just added here as comparison the activated T cells, and we call the population that is closer to the activated state the ones that are poised for activation. They're all naive T cells, but they're closer to the activated state, this cluster here, so we call it poised for activation. And then we call the other one quiescent. And so we wondered which one belongs to which, right? The hypothesis from up here is that the one with central chromatin pattern should be the ones that are closer to activation. And that's exactly what we found, right? So now we can use our model to translate between these chromatin images to single-cell RNA-seq. So literally just take this image, generate its corresponding RNA-seq profile, do clustering on the generated data, and we find that, one population lands in here and the other population lands in here. Okay, so this was the first kind of, so here it's very hard, it was very hard to do validation because we couldn't measure them in a paired way, so we actually did it just choosing two proteins and, also staying for these two proteins in together with DAPI and showed that they really separate in these two populations. Now, what is exciting now with spatial transcriptomics is you do have paired measurements. Okay, so we're very excited to collaborate here with Xiao Wang's lab at the Broad, where she developed StarMap, where you get to see, here it is Hoek staining for the chromatin, but then you also get to measure gene expression paired in the same cells. And you have another modality, because it's spatial, that we also get to see the spatial organization of cells. So now here it's paired, much easier autoencoder framework. But again, as before, every dot here is a cell. Okay, which you get by encoding, these different modalities together, where you can think of it as two cells here in the latent space are close if they're similar in terms of their chromatin state, if they're similar in terms of their gene expression state, and if they have similar neighbors, because we're now also using adjacency. Okay. Now, as usual, when you have your cells embedding, as if it was, single-cell RNA-seq, you just do clustering in the latent space. And we'd like to know that this generalizes at least across different mice, even mice that we haven't trained on, right? So all I'm doing here is mapping, every one of these cells that now is colored based on one of the clusters back into space. And you see that even on mice brains that you haven't trained on, at least, the cells go back to the same places. So they're generalizable even if you take a new mouse brain here. You can even go all the way to now here, and this is what we validated, is that you can actually predict, even from just a chromatin state, you can predict transcription, and then it's good enough to do your standard, cell type classification, identify the right markers for every one of the cell types. So all that you can do in a new mouse brain where all you're taking is just Hooke staining of the different cells. Okay, so that was just for validation. And, I put here the picture of Xin-Mi. She's giving a poster tomorrow. She's actually not talking about this work, but you'll see her picture show up a lot. But you can go ask her questions if you have any on any of these works. Now, translation and validating translation between one modality and the other is an important task. But really, we would like to use these types of embeddings, to gain new insights. This was actually in an Alzheimer's mouse model. So we wondered, can we maybe identify new biomarkers? Through this multimodal framework, can we maybe identify new biomarkers of Alzheimer's disease? So all we're going to do is, based on this embedding of the cells, we're going to predict, because that was actually also measured in the star map experiment, plaque inside the tissue. So we're going to have this prediction task where we have this embedding of cells, and we're going to predict from the embedding whether there is plaque nearby and how much plaque there is nearby. Okay, simple prediction task. And when you train such a prediction task in the latent space, you get back these, say, activation maps where it tells you how indicative is every one of the pixels for predicting whether plaque is nearby or not. So now, to me, and also Xin-Mi, when we saw these images, this is very hard for us to interpret. But actually, Shiva, when he looked at it, he immediately had a hypothesis of what the neural network might be picking up. Because to him, it was clear that, when you look at what it is highlighting, it's actually highlighting areas, not the full microglia, but actually, and some microglia, it's not highlighting at all, although they're close to plaque. But actually, it's highlighting always particular areas within the cell nucleus, areas which are heterochromatized, as now we know. But that was the hypothesis that he came up with, what the neural network is actually doing in order to predict whether there is plaque nearby. Now, once there was a hypothesis of what it is learning, well, then we can switch completely to saying, well, let's just actually figure out heterochromatin to euchromatin ratio in every one of the cells and see whether it's right. Okay, so now you have very nice bimodal distribution in every one of the cell types, just showing it here for microglia, where you can just classify, one pixel as heterochromatin, another one as euchromatin. And, now we can just look at these activations of the neural network versus heterochromatin to euchromatin ratio. In every one of the cell types, it's very, very nicely associated, meaning that this is really one of the features that the neural network is using in order to do this classification. And it's super nice, so now you can forget about the neural network. Now you just have a new biomarker of Alzheimer's disease where you see it's really significant in all cell types that with Alzheimer's, heterochromatin to euchromatin ratio goes up, which is particularly interesting because it's well known that with aging, heterochromatin to euchromatin ratio goes down. Chromatin decompacts with aging. Okay, so you have this reverse that is happening in Alzheimer's disease, and is something that we're now interested in studying, which are the parts that are, during aging decompacting, but then, of course, in Alzheimer's getting more compacted. Now, in ongoing work, we were able to now, in collaboration with Tamara and Lashley at Queen Square Brain Bank, to actually look at whether this is the case also in humans. And so we're very excited that, again, we see the same trend that heterochromatin to euchromatin ratio goes down with aging. However, and again, we took, cellular embeddings of every one of our cells. And for all of the clusters that actually show a difference in heterochromatin to euchromatin ratio, you see that heterochromatin to euchromatin ratio always goes up, significantly up in Alzheimer's disease. Exactly what we saw in the mouse models as well. Now, what we were also interested to see is to compare this to things that are known. So in Alzheimer's disease, we know that there are differences in lamin B and that we have these invaginations in the cell nucleus. And in fact, these cells or clusters where you see heterochromatin to euchromatin ratio go up significantly are in fact also associated with these lamin B invaginations. And so we really need to get to, the biology and the mechanisms of how this all relates to Alzheimer's disease. But But what is something that we're now currently looking into is if you look at these cells that have, higher heterochromatin to euchromatin ratios, or these imaginations, they're actually clustered in space. So you see here one of these clusters, cluster 7, you see how they're all localized in this part of the brain slice, and this is in fact generalizable across all of the slides that we have of these patients, whereas they're not found on this side. You also see very clear differences in how the cells are organized in these brain slides. So these disease-associated cell states are actually clustered here in particular areas inside the brain. So can we push this even further? Can we go to genetics? Can we go to protein state of a cell? Oops. What do I do? How do I get out of this? Can you remove that? Oh, perfect. Okay, it's gone. So in terms of genetics, so this is in collaboration with Melina's lab. And again, this is all work led by Shini, so you can ask her questions about also this work here. We're looking at adipocytes now. And so these are patient-derived adipocytes. And, they're differentiated in the dish. And again, every dot here is one of these cells. And now because they're patient-derived, we also have the genetic background of every one of the individuals. And so now we can start, asking questions about, instead of doing, say, genome-wide association studies at the level of an individual, we can do these kinds of things at the cellular level, trying to think about maybe this can get us closer to what is actually happening mechanistically in terms of, different diseases like coronary artery disease or fasting glucose levels that could be changed. And so now here again, so again, every one of these cells, every one of the dots is a cell. You can do clustering in this latent space. And just to show that, this latent space has it actually biologically meaningful, you see that the clusters, are enriched more and more for well-differentiated adipocytes. So there is structure in the latent space that corresponds to something biological. But now you can do things like, let me figure out what is the direction that corresponds to, say, an increase in polygenic risk score for fasting glucose, right? That corresponds to a direction in the latent space. And I can take a real cell, this here is a real cell, and I can ask if I move it a little bit in the latent space, because these are autoencoders, I can generate the corresponding image corresponding to it. And I just move it a little bit, so same cell, but now I'm generating images, these are all fake images, of how this cell would look like if it had a higher predicted polygenic risk score. And you can start to interpret what are actually the features that are associated with changing polygenic risk score at the cellular level. So for fasting glucose, you see, and we don't know yet why this is the case, but this is generalizable across all cells, is that it's actually associated with the nuclei becoming much rounder. What is maybe easier to interpret is here, coronary artery disease. Here you see a very clear change. If you walk into the direction of changing the polygenic risk score, you see a change in the mitochondrial channel. And just so that we know that we're, not making up things, that you actually find things that should be there. If we take the direction that corresponds to, more differentiation of adipocytes, you see that the main change is in the lipid channel, as it should be. Okay? But these are, hopefully we can get to a little bit something more mechanistically about these diseases if we can go like here, where we can do these polygenic risk scores inside at the cellular level. Now, what about proteins, right? So if we can really measure very accurately the state of a cell using just by its chromatin, well, if we can do that accurately, we should be able to predict where all the different proteins are inside the cell. We saw a wonderful talk by Emma earlier this morning. We want to do it a little bit different, right? Because she was trying to, and had very nice work on, when you have landmark stains together with the protein stains, to then do a very great annotation of where the proteins are inside the nucleus, inside the cell. Whereas what we want to do here is, here we don't just take the nuclear channel, but all the landmark stains that she has in the human protein atlas. And we're asking whether how much does this already tell us about where a particular protein is localized when we're actually literally going and generating the image for a particular protein. Now, because we want to, the Human Protein Atlas is an amazing resource, but really it only measures 0.25% of all possible cell line and protein pairs. So if you want to be able to predict where, and we want to make the claim, right, that from these landmark stains, this is enough to really get a very clear understanding of the state of a cell so that we can even predict where different proteins are localized, we need to be able to generalize to all unseen cell lines, cell types, cellular states, and unseen proteins. And so if you only do training with landmark stains and protein stains at the same time, you cannot generalize to new proteins. So here, in order to be able to generalize to new proteins, what we're going to use is actually the protein sequence. Okay, so two things go into the model. Cell state is represented by these landmark stains. You saw also in her talk, right? That's enough to be able to distinguish different cell types. That's enough. That's a really good representation of the state itself of a cell. But in order to also be able to generalize to unseen proteins, we also have protein sequence go in. And from these two, what we're going to generate is the image of where we're saying where that particular protein is actually localized. Okay, and as you saw in her talk, again, so this is work by Shini, so again, this is actually what she has on the poster. As you saw in her talk, if you look at the data that is measured in here, you have huge variability in terms of some proteins being localized in different places within the cell, within a cell line or across cell lines. Now, because we only measure this much of all of the pairs, well, here we can generate, right, the protein images for any protein, any cell line. So first, let me just show some validation. So this is for held out proteins and cell lines from the human protein atlas. So these are proteins that the model has never seen, and also cell lines that the model has never seen. How well you can predict where that particular protein is in that particular cell line. Now, I think always the nicer thing is you do validation in new experiments. So this is in collaboration with Fei Chen's lab. So these are completely new experiments where you see for unseen cell lines, even a cell line that isn't even in the human protein atlas, how well we can predict, even for proteins that we've never seen in the model, where they're actually localized by just generating the image of how we predict that image would look like if you were also able to stain that particular protein. Okay, so this is qualitative. So now let's go to these types of measurements of, predicting variability of, in this case, nuclear proportion of a particular protein within different cell lines. Okay, so as she also pointed out, right, there is large variability between different cell lines of where a protein is localized. And you see here that it really doesn't matter whether we have cell lines that are used in training or unseen cell lines. And here you see an example of the predicted protein that has a large variability across cell lines. And you see nicely how you really see very large variability of sometimes the nucleus being much more intranuclear and sometimes it's being much more outside of the nucleus. But also because, this model, right, I'm giving it as a representation of a cell, just the reference stains, I can predict variability within a cell line. And so this is, and again, we can do it for all cell lines, for all proteins. These are all, right, generated images. And based on the generated images, we can identify, well, which cell lines and which proteins show the biggest variability within a cell line. And so this is the one, that we predict to be very, very variable. And so here I'm showing you, the results, the actually measured data, how you see that, sometimes the protein is really very intranuclear, as it is. When we predict it to be intranuclear, it is indeed. When we predict it to be more outside, it is indeed. Okay, so this is how you can now generalize and how much information is actually just in these reference stains about where every protein is localized in that particular cell. And let me end by maybe a more challenging task. I think we've all seen really nice work in Aviv's talk on predicting the effect of unseen perturbations. We do a lot of that, but I'm actually also really excited about the opposite. And I mentioned it at the beginning where, let's take an image of a cell and are we able to tell what was wrong? what was perturbed in that cell that led to that particular image. That's a much harder task because I'm not trying to, on average, get, the prediction of gene expression correct for a particular chromatin image. I'm really taking an image, and I need to pinpoint which one was the gene that was perturbed. Okay, so that's a much, much harder task. And again, we want to be able to do that for perturbations that we've never seen. So say, I have a perturbation on, some particular gene 1. Okay, and I've never seen images of where you get that gene perturbed. But I've seen other images of some other genes being perturbed. Now, if you can get a good representation of genes, right? And, you have some images that for some genes, how they look like when they're perturbed. Now, I give you a new image, like what we show here. I give you a new image. Well, if you can map this image into where it lies in this gene space, well, then you just need to figure out which gene is close by in this embedding, and that would be my prediction of which gene was actually perturbed. And so this is the types of questions that we've been asking, just also work that is just coming out. But I think it would be really important to have more of these tasks, which are really hard tasks. I mean, I don't think we're doing really well yet, definitely better than random, but I think there is a lot more that should be done in here. Also, we show how it can be used to identify drug targets, right? So if, I give you a new cell where a particular drug was used, then you map it into this gene space, and you try to see where does it come out, to which gene is it close by, and that would be the prediction of what the target is for that particular drug. So this is kind of the reverse of going from an image to saying what was actually perturbed inside the cell. And again, here, Shini has collaborated on this work here, so again, you can also ask her for any questions that you have. So with that, let me end. I acknowledged all of the collaborators as I was going along, and you saw Shini has been leading a lot of these works. So she is here, so do ask her questions as well at her poster on Tuesday. And thank you all.
Thank you for your great talk. So I'm wondering actually what is a driving factor of prediction? So you explained about heterochromatin, but it's really difficult to believe that's the main driving factor, like as a single factor. For example, if you use decitabine, like a demethylate or HDAC inhibitors, still you can predict the same thing?
Yeah, so actually, these are really great questions. And so first of all, okay, so usually from DAPI, right, what people use and say what is captured is cell shape, nucleus shape. That's of course something, but here we really show that it is, more and we show in all these applications that just nuclear shape is simply not what gives us the signal. The biggest signal comes from, these patterns of heterochromatin and how it is organized. Now, there is a great question of like, what are things that we cannot predict just from DAPI? Okay, and we actually have this model that is on the bioarchive, again, Shini's work, which I didn't present, where we now show that we can identify what are the features that are shared between two different modalities and what are features that are not shared. Okay, and so for example, we looked at gamma H2X. Okay, and so for DNA breakpoints, we were thinking that like just DAPI should capture that. We're actually surprised to find that how many breakpoints you have and where they are is actually not captured by this stain. Okay, so there are things that we simply cannot get out of this particular stain. However, it's pattern, like the kinds of features of how the breakpoints look like, how this particular stain looks like once we know where it is localized, that's captured by DAPI. So I think a lot needs to be done by these, and we have these partially shared latent spaces where we can really go in and identify what are features that are not captured by a particular modality and what is captured. And we need much more of that. So I cannot tell you for your particular protein, what is captured and what not. But we have the models to be able to test it.
Also I do spatial transatomics, and like a star map I'm familiar with. They, in general, do hydrogel before measurement. And based on my experiment, actually sometimes genomic DNA shape changes after hydrogel the protein scale.
Yep.
So that kind of thing is accounted?
Actually, so the nice thing about doing these multimodal, and again, there is other work that is ongoing that I didn't show, There is, as you're saying, like the preparation of like the particular tissue will matter a lot, will lead to batch effects in transcriptomics. But because what we're looking for here is only what is shared between transcription and these, say, Hook stains, for example, or DAPI stains, actually the batch effects that you have in these stains are different than the batch effects you have in spatial transcriptomics. And they're actually removed. So we're able to even generalize to, we looked at FFP and fresh frozen slides. And we can even generalize across like very different things. We have an ovarian cancer data set where in Australia they generated a huge amount of data. We were able to generalize to that data just using H&E. Because H&E has very different batch effects at spatial transcriptomics. Those are the things that you get out for free when you use these multimodal autoencoders.
Thank you.
Yeah.
Hi, Caroline. That was fantastic. I'm glad you left this slide up because my question was sort of thinking about expanding further. Given the examples you gave with microglia and Alzheimer's disease, really leveraging the spatial data, transcriptomic, and now this sort of shift from heterochromic to euchromic ratio. One of the things now, I think the challenge is then to take that one dimension, looking at the nuclei, looking at the chromatin, and then now we do have data on multiple other modalities on what actually that means for the biology of microglia around a plaque or with different challenges. And I think one of the questions of how you then go from this type of data to a biomarker, a non-invasive biomarker. What's really cool about Alzheimer's genetics is it's nominating immune cells and myeloid cells across the body. So I was wondering how you're envisioning like leveraging some of the IPS models, some of the other types of analyses that are actively being investigated to then bring other types of data on top of the chromatin to really understand like what that activated state means and how we might be able to read that out in biomarker at the proteomic or other level from the peripheral tissue.
Yeah, I think these are great questions. So first of all, now in the human, we do have all kinds of genetic data in addition. And so we already showed that these things are, what distinguishes different genetic states, et cetera, that we already know from in these neurodegenerative disease contexts. But we want to go more into the details of what is functionally happening. And so, how we see it is actually really this protein. If we can really predict protein subcellular localization and how that might change, right? So we'll take like one of these cells that has a high heterochromatin to euchromatin ratio. All we're putting in is, even if you just put in DAPI, you can predict the protein localization and look at which ones of the proteins are differentially localized. That's why we're so excited about this work, which all it takes in is these stains.
Right.
This is the representation of cell state, and from it, and protein sequence, and from it we generate the image of how that particular cell looks like for the different proteins.
Yeah, I would just say the IPS models where you can then also do cell paint and look at other dimensions on top of that could give you like another level of sort of functional readout.
Yes, and that's exactly what these are the IPS models in the adipocytes. This is exactly what is used here.
Thanks.
Yes.
Okay, I think we'll end it there. Thank you again, Caroline.
Thank you.